Good morning, everyone. It's 30 past 11, so I propose to start. Thank you for joining our third edition of the Tuesday Connect. So before giving the floor to Lucie and people from Work Package 8, just a few reminders about Tuesday Connect. So keep in mind that this meeting will be recorded, and that the recording will be made available on YouTube after the meeting. So if you don't want to be recorded, please turn off your camera and post your question, comments in the chat. For helping us moderating question, please add your name and institution in your name tag. For convenience, please mute yourself when not speaking and raise your hand if you want to intervene. And you can ask questions to the people who will present. So you can do it in the chat or just raising your hand after the presentation. And if possible, please turn on your camera when speaking. So now I will give the floor to our colleagues from Work Package 8 who will present their work on task 8.2, animal health, and 8.4, integrated One Health surveillance. So Lucie and Work Package 8 colleagues, the floor is yours.
Hello, good morning, everyone. Good morning, Johan. Can I share my own screen?
Yes.
Okay, so I will try. You will tell me whether you can see it. Let me move to full screen, maybe. Okay, can you see my screen now?
Yes, perfect, Lucie.
Okay. So let's start. Good morning, everyone. My name is Lucie Collineau, and I'm a veterinary epidemiologist at ANSES, the French Food Safety Agency. And together with Sophia Ni from the Public Health Agency of Sweden, we are co-leading the Work Package 8, which is about improving One Health surveillance of AMR in Europe. So we are working and talking today on behalf of a large group of 25 partner countries. And our main partners and stakeholders are national AMR surveillance experts, which are based in national governmental agencies, expert laboratories and ministries, working in close collaboration with EU and UN agencies. Our overall objective is to reinforce the European strategy for One Health surveillance of AMR. And specifically, we aim to strengthen the national surveillance system on AMR in the human health sector, achieve a full-scale implementation of the European AMR surveillance network in veterinary medicine called EARS-Vet, set up a European AMR surveillance network in the environmental sector, EARS-Env, and develop a national annual One Health surveillance report in each participating country. We work under the umbrella of the Council recommendation on stepping up EU action to combat AMR in a One Health report, and there are 6 specific recommendations that are related to surveillance, and these have been translated into national action plans. And earlier this year, there was a framework that was released to evaluate the implementation of those recommendations. And these have been translated into outcome indicators and output indicators that we are also aiming to inform or support with our current activities. So you're probably aware that a lot of progress has been made in the recent years to improve One Health surveillance of AMR in Europe. And a very good example of this is the JAMRAI report, which is a joint integrated analysis of consumption and resistant data in human and animals. So what ECDC, EFSA, and AMR are doing, they're joining forces to look at statistical association between consumption and resistance within sector, so these are the vertical lines here, and between sector, which are the horizontal lines on this figure. So this is quite an advanced level of integrated analysis of AMR between sectors, but there are also limitations in the way this JAMRAI analysis is being performed. First, the data on AMR in humans only focused on diseased animal with data from EARS-Net that focus on bloodstream invasive infections. But this is only like the tip on the iceberg, and there are other infections with resistant bacteria, which would also be considered. In the animal sector, data included in this integrated analysis originate from the EFSA monitoring report on zoonotic and indicator bacteria in healthy animals, but we have no data from a diseased animal that could be used to compare with AMR data in diseased humans. Third, an important gap, obviously, is environmental surveillance, which is missing in this integrated analysis. And fourth, we think a gap is that, okay, this integrated analysis is performed at the European level, but there are no similar, in most countries, no similar analysis being performed at national level. Some countries in the EU have a One Health surveillance report, but often it's not super integrated. So you have like chapter 1 human, chapter 2 animals, and sometimes chapter 3 environment, but no really integrated analysis of this data across sector. So these 4 areas are those we're trying to cover with our work package 8 activities. And for today, the focus will be on the environmental and disease animal activities. So these are performed as part of 8.2 and 8.3. And then the thematic for One Health surveillance, so integrated analysis and human health surveillance will be presented at the April thematic webinar. So for those more interested in this aspect, hope you will also join us in April to know more about what we're doing for the cross-sectoral and human health surveillance. So with this now, yes, so these are the tasks, sorry. So we'll focus today on 8.2 and 8.3, and then in April we'll cover 8.1 and 8.4. So I will now give the word to Peter. We'll start presenting about our animal health activities and present the current progress and future plan for EARS-Vet surveillance of AMR in diseased animals.
Okay, thank you, Lucie. And yes, my name is Peter Damborg. I'm a veterinary microbiologist from University of Copenhagen, and together with Lucie, I'm task leader of 8.2, the part you can see here on EARS-Vet. So, and I think, Lucie, can you move the slides for me, please? Thank you. So, there's, as Lucie already mentioned, we have surveillance on AMR in animals already. We have in the food chain, we have indicator E. coli, and then we have surveillance of AMR in zoonotic bacteria like Salmonella, Campylobacter, existing already from EFSA. So, of course, this is important information being generated on foodborne AMR transmission, or the potential for foodborne AMR transmission. But as also pointed out before, we don't have any data on clinical isolates covering, of course, both the food animals and companion animals. Next slide. And this is, of course, a problem in the view that we know AMR exists in clinical isolates, because, of course, we have some knowledge already based on publications, based on national surveillance. So there is some, you can say, scattered evidence of emerging antimicrobial resistance in animals. What you see in this slide is examples of carbapennemases in both food-producing and companion animals. But of course, we also know of other emerging AMR problems in animals. And yeah, this is something we should try to cover better at a European level. Next. So therefore, as part of the first edition of EU-JAMRAI, this EARS-Vet initiative was established at a small scale, you can say, compared to what we have now. That I will get back to. And the ambition was to try and parallel the existing EARS-Net network in the human sector and try and close some of the gaps that I mentioned before. So have some AMR surveillance at European level in clinical isolates from animals. And there are several reasons why this would be a good idea. You can see some examples listed here. Apart from just complementing what we have already, this could be used to support antimicrobial stewardship initiatives. And there are, of course, many initiatives encompassed by this. One of them could be development of veterinary antimicrobial guidelines. There are various initiatives in the recent years to develop such guidelines. And in order to recommend antimicrobials, of course, we need to know levels of AMR. Supporting risk assessment, including the risk of zoonotic transmission. Advising EU-level policy making. So instead of having, you can say, opinion-based, expert-based decisions on policies, and legislation and so on antimicrobial use, would be nice to have some evidence base before such decisions are being made about restrictions or other things. And then, of course, if interventions are established, it would also be nice to see, to be able to compare before and after what is the level, and compare between countries in a harmonized way. Next slide. So the next few slides, I will just briefly go through the main, some of the main achievements from this first EU-JAMRAI project. And one of the things that was done initially was to map existing national surveillance systems of AMR in clinical isolates. And what you see on the left here is this map that came out of that. And you can see the green countries are those that had an existing AMR surveillance system. And the other colors, those that either did not have or for which we didn't find out if they actually had something. And even if we had, even if countries had existing AMR surveillance systems, they were very diverse, meaning it could be a centrally coordinated AMR surveillance system from a national agency, could also be different private labs from which AMR data was collected and then gathered and possibly published also. So it was you can say, quite diverse landscape we identified. Next slide. Another thing was to try and define a scope of EARS-Env. So essentially to find out what should EARS-Env be about, which animal species, which bacterial species, and so on. And this was made in a bottom-up approach where we found out some of the countries having an existing AMR surveillance network. What did they actually monitor? What you can see in the right here, you can see how many countries had surveillance of AMR in cattle, in different animal species. So based on that, and based on expert opinion, we did select which animals, which bacterial species, but also which antibiotics to cover in a proposed future EARS-Env network. And then it was decided that interpretation of antimicrobial susceptibility testing should be done using ECOFs. Next. One thing is to talk about EARS-Env, and one thing is to have some nice ideas. But I think the most fun part of this was actually to do a pilot study. So this was done with the partners you can see on the left here, also indicated on the right. So quite a few of those with an existing AMR surveillance system, and then actually also a few other countries for which we were able to obtain data. Next slide. So this was published 2 years ago, the outcome of this pilot study. So you can see here how many countries, 9 countries, 11 partners, data from a 4-year period, and actually quite a few isolates all this was based on. And what you can see on the left is an example of the AMR data published. I will not go into detail, but just so you can see how it was presented. E. coli resistance to aminopenicillins in 6 different countries from which we had the data. And you can see here, each color represents one animal species. So as this should be clear here, it was a bit diverse what we could find. Some countries had, well, some of them not presented here did not have any data. Others had from one animal species, and other countries again from several animal species. So some of the strengths, of course, we had access to raw data, MIC data, or distribution data. So we could interpret it in the way that we wanted to. Of course, the costs relatively low for obtaining the data, but it took a while to analyze the data. It was a bit complex to try and analyze it with all the different formats of data we received. And then we can talk about the representativeness and the volume. I will show in the next slide. It was not ideal. Harmonization, as I mentioned before, it was not just one method. It was both MIC data, and then from France, we had distribution data. And an interpretive criteria we had for quite a few of the drug-bug combinations, but for some of them we could not interpret anything because there were not any ECOFs existing, or simply because the range of MICs being tested were not sufficient to interpret according to ECOFs. Maybe they were tested originally for clinical breakpoints, but this time we wanted to do it different. Next slide. Yeah, this is just a table showing the origin of data, of isolates. And I think it should be clear here that it was not, you can say, harmonized between the countries participating. France, the isolates from France constituted almost 80% of the data. So, of course, this was a first attempt. I think it's interesting what we found out, but we also learned a few lessons on what could be improved. Next slide. So in JAMRAI 2, we want to make everything bigger and better. So we want to include more countries, more isolates, and so on. And then we want to do it in, you can say, a more systematic, harmonized way. So in this, we have different subtasks where we aim to collect data in a more harmonized way, and also make it easier to collect data and analyze and report data. There are also initiatives, I will get back to those, on whole genome sequencing. And then we want to try to integrate what we are doing with the EU AMR surveillance landscape. Next slide. So next slides, I will cover a bit what we have been doing in the first year and a couple of months actually now, and also explain a bit what the plans are ahead. But for one of the subtasks, the most important one, collecting data, we have started by updating the scope. So the scope I presented before with the animals and antimicrobials and bacterial species we have looked at, and we have decided to extend it a bit. So we cover now more animal species, including horses, sheep, and goats. And also we have extended the number of bacterial species, and we have looked at the antimicrobials being tested. Then in order to facilitate data collection, a database has been developed. A WhoNet software module has been developed in order to facilitate the collection of data and make it easier to, well, both collect, submit data, but also analyze the data in the end. A data sharing agreement has been signed, so to ensure that we don't do anything wrong. And then we have just last month opened a call to receive data, MIC data and distribution data. Next slide. So for harmonization, as I mentioned, this is an important part with different methods being used and so on. We're in the process of drafting a manual covering both the bacterial species, the scope, and as such. how data should be collected, the methods we accept for laboratory analysis, and so forth. So essentially, explaining what we expect from the partners and what we are going to do with the data. And of course, this is something where we need to update it on a regular basis to see if we can, especially after we deliver the first data, to see if something can be adjusted. We are developing MIC plates, customized MIC plates, in order to do proficiency testing between labs and see to again test if data are comparable or not, but also for with the possibility for future diagnostics. Ideally, each country should or all the countries should use the same plates, the same methods for susceptibility testing. So this is, you can say, something we are planning now, but probably it will take a while before all countries use the same methods. At least we are aiming to try it out. Next slide. Whole genome sequencing is another subtask where a subset of isolates would be selected for this. We have a budget for 500 isolates, and we are aimed to look at some of the AMR phenotypes you can see here, including ESBLs, carbapentamases, and MRSA, to map what we have and to try and see if some of those overlap with what is seen in human medicine, and simply to try and follow what is going on about potential transmission in Europe. Yeah, and then integration with the existing EU AMR surveillance landscape. We have established already collaboration with different initiatives. You can see in organizations, EFSA, FAO, INFARM, and then the EU partnership on animal health and welfare, SOA8, that I'm sure some of you are familiar with already. Also looking at surveillance, and in this particular case, especially the part of SOA8 focusing on development of ECOF, so new interpretive criteria. Next slide. So this is just a timeline. I think I've explained most of it already, that we launched data call. We are developing MIC plates, and we are developing the EARS-Env manual. All of this should be ready this year. And the data collection we should have ready this year, and then hopefully at least some idea of the AMR landscape in animals across Europe by the end of the year. We are planning a meeting in Copenhagen in May, and then we are planning what should happen ahead. Of course, based on the outcome, we should produce a report. We have some other ideas on how to present data, publications, dashboard, etc. And then, as I mentioned, try to integrate with the existing AMR surveillance across Europe. And then you can say the most important thing, or one of the most important thing, is sustainability. One thing that we care a lot about, because it shouldn't just be an initiative launched during a 4-year period of JAMRAI. This should be something that could hopefully continue beyond that. So this is something we focus a lot on and try to collaborate with different partners and see if we can somehow make it a sustainable initiative. Yeah, next slide. I think is my last slide. This is just an overview of some of the outcomes from the first JAMRAI project. So 4 publications, essentially with more detail on what I just mentioned before. So if anyone has any interest, you can read more here. Next slide. And this was it from me. This is a picture from the first physical meeting we had in Paris last year. And if anyone, if you have questions, of course, I'd be happy to answer them. I'm not sure if we should do it now or at the end of the seminar, but otherwise you're more than welcome to contact me in this mail address or Lucie afterwards. Thank you.
Excellent. Thanks a lot, Peter, for this presentation. I don't see any question in the chat right now. Is there any question from the audience?
You can also raise your hand if you want, if you have a burning question.
Otherwise, we can continue, and then if there are any questions at the end, we could take them then.
Mm-hmm. Okay, let's do that. And as Peter said, we're also available by email for follow-up if needed. And also, Johan has mentioned in the chat that we'll make these slides available to the participants. Also, this meeting is being recorded, so we can also, share it later. Okay, so thanks a lot, Peter. And if there's no further question, we can move to the second presentation. And I think you're sharing your slides, right? Page 3, colleagues. I don't know who's presenting. Or do you want me to do it? Please let me know.
Sorry, we had to find the unmute. Do you see our screen right now?
Yes,
Yes.
we see it.
Perfect. And we'll continue. Welcome everybody to the second half of this presentation. We are going to talk about surveillance of AMR and the environment. So not about integration, but today it's the environment. Happy to be here together with Roosmarijn and Luiken to my side, and later on Mark Montforts to report on what we have been doing in the environment. So first of all, to set the context, we are situated a step back from what we know about veterinary and human surveillance. Actually, while we know a lot about the presence of antimicrobial resistance in the environment, we know that we can find resistant bacteria, resistance genes. We can also find resistance mechanisms in pathogens in different matrices in the environment. We also know that anthropogenic pressures can affect AMR dynamics. Most importantly, emissions with human wastewater lead to the contamination of the environment, but also emissions from animal manure. Resistance can also occur naturally in the environment or be triggered through the presence of substances like antibiotics. So there's a lot of dynamics around the environment. And last but not least, humans can also be exposed to resistance as it is present in the environment. For example, through recreating in surface waters or through other contact with environmental media. So in this context, what we think that environmental surveillance can contribute is to understand better how AMR spreads from humans and animals to the environment and within the environment, and how that then reflects back on risks to humans and animals through environmental exposures. Unfortunately, though, there's a lack of harmonized approaches how to conduct such surveillance. There are some national experiences, often from the most recent years. There's a couple of international documents, the WHO Tricycle Protocol, but generally spoken, there's very little advice on how to conduct environmental surveillance. And that is in contrast to the call for integrated surveillance that has been expressed, for example, in the Council recommendations, but also by the United Nations General Assembly. And that makes JAMRAI to be one of the fora where such a common surveillance strategy addressing the contribution of the environment to AMR in the One Health framework could and should be developed. So that's the goal we have ahead for the coming years. And the ambition here is to start well, and develop that into a harmonized and well-structured process. To this end, there are actually two ambitions. First of all is indeed to develop such surveillance, so use what we know from scientific research, from some governmental activities, and develop that into an action package by jointly determining objectives, sampling strategies, and indicators for such environmental surveillance. Surveillance that could then be taken up by countries when they develop their national action plan actions and could help them with thinking about the environmental compartment. This can also be employed in current and future European regulations, as there's a current development, for example, within the Urban Wastewater Framework Directive and also within other directives to more completely address also environmental dimensions of AMR. Similar to EARS-Env, we also want to establish a network, an environmental AMR surveillance network. Why is that useful? Because we see that many countries are employing their first steps in environmental surveillance. So we try to bring a community of practice here, and we also endeavor to integrate with other sectors and networks where possible and where needed.
Yes, and here I take over. As Heike was saying, we're in quite a unique position to really think about this surveillance from, well, maybe not scratch, but from the beginning. And I'd like to take you through some important steps in the design of surveillance, and in our case, environmental surveillance. And we really feel it's very important to start with the definition of the objectives, as there can be several objectives. So we need to agree with each other what we're aiming for. And in general, the four main overall objectives are detecting AMR trends in the environment. And a special case of environmental surveillance is AMR trends in humans through wastewater-based surveillance. Another objective could be the emergence of new forms of AMR, or study the efficiency of interventions. And last, provide data for risk assessment. Then we need to define some sample strategies. And important to say is that the environment is, of course, not one thing. So we need to think about approaches for environmental sampling in several different compartments or matrices. For sure, water, soil, wastewater, and several others. And if we know what our objective is and in which compartment, for which compartment, then that's the right basis to think about the common indicators we want to measure. So indicators, we mean AMR agents that you can measure and the methods that we can use for that. And it's important to stress again that here we really need collaborations with the other domains, other stakeholders, as most of these objectives are to serve human and animal health. So there we need the input from other sectors. And of course, we hopefully create, and it's needed to create a support network. So what are we more practically doing in JAMRAI? Several things. First, we are collecting information of experts and of countries, of different projects, to 2 different questionnaires. About that, I will elaborate a little bit next slides. And to create our own vision with our group of experts and everybody working in the environment. What do we think a good environmental surveillance looks like? Another important step is consultation with other stakeholders and also alignment with other European projects and regulations. And we are very happy that we have the opportunity to do a pilot within JAMRAI. And in this pilot, we will test a common approach to environmental surveillance and some central elements. And lastly, and that's something that Mark Montforts will elaborate more on in a few minutes, is explore what we need to know more about the usage of antimicrobials outside of that what is regulated within healthcare. So in this presentation, I will talk a little bit more about this part and what we are doing, and Mark Montforts will talk about this part. So as I was saying, we have drafted 2 questionnaires, one for mapping current surveillance initiatives in the member countries, and the second one is to collect visions of others, of experts, of countries, of researchers about potential future surveillance in the environment. And here you can see a list of several things we are asking in the questionnaire. And we are aware it's a long and quite complicated questionnaire. But we are very happy that the answers are coming in. The deadline is the end of March, so we do not have all responses yet. But we are happy to say that for mapping the existing environmental surveillance activities, we now have 17 responses, and for sure more are coming. And input from the different countries and different domains on vision for future environmental surveillance is also coming in. We already have 10 responses, and for sure more will be coming. So this is from others and input from others. But of course, we also want to create our own vision for future environmental surveillance and a common approach. And this is what we're doing in partly in collaboration with another initiative, EU-WISH. That's a joint action focusing on surveillance of wastewater in Europe. And as we are faced with the same challenges of setting objectives, of setting indicators, we feel that it's very important to join our forces, join our work. And we had an initial meeting, so everything is started. And I'd like to take you through what we will be doing very practically, and we'll be doing this this afternoon. Actually, we have a big meeting with all involved WISH and JAMRAI people, and this is exactly what we're gonna talk about. And as the different objectives that I was just naming, and the different options for environmental compartments. And we all ourselves are gonna prioritize compartment plus objective. We're not gonna form an objective and a plan for now for every compartment and every objective. No, we want to prioritize. Is it determining trends in raw wastewater? Is that our first priority? Or is it determining emerging forms of AMR in recreational waters, for example? So these discussions are going on, and this afternoon we will have that together. And if we have prioritized this, then of course the next step is prioritize indicators for a certain compartment with a certain objective. And in theory, there are just many options for indicators. And that's why we want to have these discussions together to see if we can come up with a priority list. Yes, and with this, we follow the steps that we are also asking in the questionnaire to all the countries and all the experts out there. And so hopefully together, this will be a nice basis for the common approach that we hope to have identified in June. As you can see here, these are our next steps in the coming year. So in April, we will have the outcome of the questionnaire. In June, we will write down a common approach to environmental surveillance. And in the fall or winter of this year, we will have hopefully the preparations ready for a pilot and start a pilot on environmental surveillance in the member countries in our group. So Mark, over to you.
Thank you, Roosmarijn. Okay, we'll give some details on what we're working on in this specific subtask on disinfectants and fungicides. To look into the feasibility of collection of data on usage. I'm Mark Montforts. I work at RVM. I'm an environmental risk assessor. I have been working on pesticides, biocides, veterinary medicines, fertilizers, and AMR in the environment. And I'm leading this subtask. Slide. Just for the last bullet, this information on products and disinfectants and fungicides is of course needed to get a grip on cause-effect interventions. I also showed a picture of the book that I edited 15 years ago to show you I do have some affiliation with resistance. Within the work package 8.3, therefore is this on the blue that we're going to explore what we need to know about uses of antimicrobials, especially outside the animal healthcare and healthcare sectors, support diagnosis and interventions. Slide. This task is planned to run until 2027. We have already started drafting fact sheets to get a grip on the substance, the matter here. But we're working towards recommendations on a common environmental surveillance. Slide. So what are we talking about if we talk about disinfectants and fungicides outside the veterinary medicine sector? But there are a lot of products on the market that contain disinfectants, antimycotics, fungicides, bactericides, preservatives, so they have antimicrobial properties. And here's a small list. I think these are the main ones covering is the pesticides, the biocides, medical devices can contain antimicrobials, or there are products to disinfect medical devices, and these are also medical devices. There's food decontamination practice, there are feed additives, and there are food additives. And also in cosmetics, we can find preservatives or otherwise antimicrobial substances. So within our working group, we are well, writing down what is this and how big is this sector, how is it used, and our data actually collected. So we get a better understanding of what does this environment, this chemical environment look like, right? So the problem is, you could say that we would like to have data on what is sold and how it is used. And the problem is that there are EU data on EU level for plant protection products, but for all the other ones, there are basically no centralized EU data. Well, that may not be a real problem if it's not really substantial, but we know from a joint report by EFSA, AMR, ECHA, JRC, ECDC that the use of azole fungicides in crop protection is about, in volume or mass, 1,000 times higher than the use in healthcare. Well, big numbers always are important, but then of course we say as scientists, but where and when and how is probably more relevant than just your total use, isn't it? That's exactly the point. We need to know where, when, how, and what conditions substances are used if they actually create a problem, right? So this is what we were trying to get a grip on. What kind of data do we need? We have to come to terms with our own data needs. So how, what is actually the status of a product? What is the total use, but also how it is used, where it is used, what dosages, what kinds of durations, and all kinds of metadata, descriptive statistics of the types of use. Do we need acreages? Do we need geographical patterns? Do we need trends in time? Numbers involved, like consumers, patients, animals, the different sectors like healthcare, like animal, livestock. And of course, I think the connectivity to the animal or human health resistance risk factors. We want to know what is happening where it happens, not where it just is there, nothing happens. Right. So we need this data, then you have to go back. You can want everything, but that is going to be a hard job. So it's an iterative process of really understanding what do we need this data really for? So what do you really want to research? Just calculating large numbers, like that we actually sell 30 billion DDDs per year. It's impressive, but it doesn't help. I think B and C is supporting the causative link between use and development of resistance and identification of intervention measures. That's what we need the data for. That brings us to number 4, the questions. Well, what kind of research would you people need? What kind of data? Especially, of course, when you work inside the EARS environment network, but actually also outside. If you see like, oh, I've always been focusing on the medicines, but is in my sector actually biocidal use also of importance? That's where we need to find crossover. If you have any ideas or questions, let us know so we can enlarge our network. Right? So we are thinking that the work will bring us to recommendations. I underscore this again, it's recommendations on what kind of data should be collected. And we bring into that all kinds of aspects that need to be considered to make it actually feasible, because there have been trials and errors on collecting data. This idea of what are needs and what is most likely and how could it work is a very important first step towards a real data collection plan. But an integral data collection and management plan is not what we are going to do. So don't worry, there will still time to improve at the end. That's just to manage all the expectations. But we do need input when it comes to understanding what is the data need, what kind of level of detail, you could say, is really pertinent to making the next steps. I think another slide, or is this it? This is it.
Thanks. Thank you, Mark, for this excellent presentation. Is there any question from the audience or in the chat for the speakers that presented today? Okay, I don't see any. Oh, there's one. Yes, Oksana, do you want to?
Yes, I have a question, and it's about environment, the last, and we in Sweden have also, of course, one health approach when it comes to antibiotic resistance, but we have had a challenge to incorporate the environment in our projects. So which challenge have you found in your projects? Have everybody been on board straight away, or have you faced challenges where maybe institutions who work with environment have felt like they're not, they don't understand the rule? Can you share something of that kind of experience?
Yes, of course. I think that hugely depends on in which institution you are and in which country you are. I think we are in the Netherlands in a quite lucky position because our public health institution, the RIVM, is also the relevant institution for actions on the environment. So actually, we have all sectors in one house, in one building, in one institution, and that makes it much easier to reach out to our colleagues, but that also makes it much more intensive, the dialogue with our colleagues. And I think that is, in our experience, that's quite needed in order to always gauge and validate whether what we can provide as environmental microbiologists makes sense for the human and animal sector. And for many countries that we collaborate with, we see that this is quite some challenge often when industries are not in intense contact, and this contact needs a while to establish and bring forward the questions that do really matter. What do we need from the environment? What is your interest in environmental exposures, and how can we feed you that data that you actually need? Yeah, and I think it needs time, and it needs an overarching One Health institutionalized mechanism body. That's, I think, what can often help.
Thank you.
There's somebody
Yes.
who raised their hand.
Hello, can you see me, hear me? I'm sorry.
Yes.
I'm from Norway, and I'm a part of the ESAC net. That is, we are working on use of antimicrobials and also we have data on the antimicrobials in animals, and we have collected now a database on the antifungals and put it together. It's just a start, but I guess that several countries can do the same if you collect the data from different parts. Would that be interesting to look further into? This is a question to Mark, actually.
Yes, I mean, it is good to know that these data exist, are being collected, because it also then brings up the idea like, how are we going to manage these data sets? If you compare it to the protection products, it's like data are being collected for the whole of the EU every year. And that is, it's in the hands of Eurostat, so it is actually governed as a goal data selection system, and it's something we have to find out if that is needed in the first place for the real questions you are asking, you want to research, but if it is needed, then the other point is, of course, like you say, you have collected data, for example, on antifungal use, that is maybe for a year, certain sector. Where do we keep and store these data so that we actually can find them? There's a challenge, but that's what we're trying to figure out.
And also to know whether it's other countries doing the same things. I know there's this ASOL network, but I mean, it's all these other types of antifungals that's out there. And I don't know the effect on humans nor animals on all the agricultural antifungals that exist, which is a bit difficult to grasp. Yeah.
That's I would like to research that, but I need data on. And well, those are the steps that will identify what kind of data do we really need. Next steps will be, how do we actually make this work? How would the initiative be?
Any other question? No, so I think we can wrap up the meeting. I think you all deserve your lunch. So I just have one slide to finish. So just to remind you, the next edition of our Tuesday Connect, which will be on April 8th. So it's a bit earlier than usual, but it was to avoid a conflict with the ESCMID. So we will meet on April 8th, and you will have the continuation of the story with a focus on human surveillance and integrated surveillance. So, I see. Oh, no, it's Richard. Okay. So yeah, thank you for joining today, and looking forward to seeing you in April.